All right, so I'm going to call this Getting What We Want, right? We've done a whole bunch of work right now. So things we've done, we designed a plasmid. Right? We made our plasmid. We transformed bacteria. Right? So the question is now what? So before I would take what I grew, I want to make sure if it's what I want, right? So getting what we want. So part of this is going to be harvesting our plasmid. So we're going to say harvesting our plasmid is what we're going to because we have to confirm it's what we want before we can move forward. Okay, so we're going to harvest it. So plasmid purification is where we're going to start, right? So this thing right here, plasmid purification. So on a small scale, we're going to call it mini prep. So mini prep is a term that's used to describe purifying plasmids from bacterial cultures. Okay. All right, so it's the process of growing selected bacteria in a liquid culture and then purifying the plasmids. All right, so that's what we're going to do. We're going to grow this in a liquid culture, and then we're going to purify our plasmids out of the bacteria, right? And if everything works, right, then we're going to amplify our plasmids for further experiments. Okay. All right. So our liquid cultures must be warm and full of nutrients to be oxygenated. So I'm going to grow it. I'm going to grow it in a Schenke incubator. So if I had a tube, right, so if I had a test tube, right, and in my test tube I had broth, right, if I just grew it in that test tube, all my bacteria would sink to the bottom. And we know from E. coli is a facultative aerobe. So E. coli grows best up at the top, right? But it'll be sinking to the bottom, so it will grow, but it won't grow well, right? So I need to make sure it's shaken and it's full of air. So shaking incubator, right? So it shakes both sides. I got one in the back of my room. Right? They're super expensive, but they work super good. And as it shakes it, it will cause agitation and it will cause oxygen from the environment to circulate through that tube, right? So I'll use a shaking incubator to grow it. Now we're going to harvest it between 16 and 24 hours. So if you look at it, 16 is right about here, and 24 is going to be over here. So we're going to harvest it when we have the maximum amount of product, not the maximum amount of growth, right? We're going to harvest it when we have max product and not max growth, right? Remember, if I harvested the bacteria for doing transformation, I'd want it between 6 hours and 12 hours, right? So if I was going to do transformation, I want it between 6 hours and 12 hours in the log phase. But I don't really want log phase here because I'm not transforming, I'm harvesting, right? So I want it at the end of the stationary phase. That's where I want it. And then I'll go ahead and purify the plasmids out of there. All right. So between that 16 and 24 hours, right, I'm going to take my bacteria that's in my LB broth tube, right, and I'm going to centrifuge, right? So I'm going to take my LB broth, right, here I'm going to change my colors here. I'm going to take my LB broth and I'm going to microcentrifuge it. All right, so that microcentrifuge, remember microcentrifuge is going to separate by gravity. So if I take my tube, my microcentrifuge tube, and I had LB plus bacteria, right? I'm going to centrifuge it, and all the bacteria that was mixed in here will form a lump at the bottom. So a couple of things. So this is going to be called a pellet. And everything here above it, we'll use a different color here. Everything above is going to be the supernatant. Right, so it separates by mass using gravity. So centrifuge will spin it really, really fast, and it does all the heavier stuff will go to the bottom. All right, so I'll spin it. It moves the bacteria to the bottom. Right. What I'll do then is I'll remove the supernatant. Okay, so I'm going to spin it, and I'm going to remove the supernatant. Right. So that's what I say here. So the supernatant is going to be removed. And it's going to be replaced with an alkaline lysis buffer. Right? So I'm going to use SDS as my common buffer. Sodium dodecyl salt, it stands for. But I'm going to use this alkaline buffer. Right? It's a soapy solution. And what that's going to do, it's going to denature the plasma membrane. Right? And it's going to cause it to come apart, and then I'll just have the pieces. Right? So if you look at it here, so look at our thing. So we're at this step right now. I put my alkaline buffer. Whenever you see this right here, then I'm going to centrifuge again. Right? And I'm going to get my precipitate again. Right? So at this point, I'm going to remove the supernatant again, right? So what this SDS is going to do, right? So this is going to dissolve membrane. And I'm going to make ssDNA. And this SS right here stands for single-stranded. So it's going to make the DNA come apart, right? And we'll spin it and we'll put it all to the bottom, right? And then I'm going to remove the SDS. So I'm going to remove the supernatant. And then I'm going to, my next step, I'm going to neutralize so we'll say we're going to use a weak acid, right? Right? A moderate to weak acid. We're going to neutralize it with what we call a clarifying buffer, right? Right. So what that's going to do, this is going to allow the plasmids to reanneal. Now the gDNA won't reanneal so well, and so when I spin it again, so then I'm going to centrifuge again, right? And the gDNA is going to end up in the bottom of this tube with the proteins and whatnot, right? Then I'm going to remove the supernatant again, right? I'm going to put a wash buffer in, and then I'm going to spin it again, right? And then I'm going to remove the supernatant again, right? Make that a different color for you, right? And then at that point, when I remove the supernatant again, I should have mostly plasmids, right? The plasmids will be mostly in the supernatant. So, all right. So let's go here. So the pH is neutralized, right? Allows for reannealing allows for re-annealing of the plasmids, right? So I centrifuge again to remove the heavier debris, and the G and A get stuck at the bottom, right? And then I can purify to remove salt. So I can do alcohol precipitation, right? Actually, propanol would be better. So what this does is it removes the salt and it causes the gDNA or the pDNA to clump together now, right? So I can precipitate that out and remove the supernatant. And at this point, I have plasmids. So we'd be doing mini prep in class, right? So this has plasmids. Now, this procedure right here is very basic, right? We'll do it better. So the problem with this is at the end, I'll get my plasmids, but I'll still have other contaminants in there. So it doesn't do the full job. So one of the ones that we use in our class, oops. All right. So what I want to do is I want to make it a little better. So let me make this a little bigger. Okay. So I'm going to use chromatography, right? So you may remember in biology doing chromatography with a piece of paper, and you made a little line on it, and you put chlorophyll on it, and you separated the different pigments, right? So that's a different type of chromatography. So chromatography, right, is a column used to as a differential filter, right? So I can filter certain things and not other things. The column can filter by size, right? So it can be a molecular sieve, right? Sieve means filter again. I can filter by charge by using charged beads, right? So for DNA, right, I can use a positive resin beads. And because DNA is negative, it will stick to these positive resin beads. I can use polarity, right, nonpolar and polar, or I can use affinity by conformation. I can use antibodies to seek out things. So we'll talk about all those a couple chapters later, right? But I can use chromatography or column chromatography to better separate my plasmas, right? So the silica gel acts as a filter medium. In industry, we'd use what's called HPLC, which is high pressure liquid chromatography. So our little filters we use fit inside microcentrifuge tubes. They're really, really small. But I could have a column that's a meter wide, right, in industry, right? And they would put it under pressure, so they would just push everything through, right? So HPLC centrifugation is used to speed up the process. Different buffers are used to either bind or release. So like I said, so here we go. So let's talk through this really quickly. All right, so this is after I put my LB in, right? And then I spun it, I got my pellet, right? And then I'll add my SDS buffer here. Right, and I'll resuspend my cells. So we haven't been in labs, so we've missed a whole bunch of stuff. So I'll use a vortexer. You might have seen those in the Labster videos. I'll use a vortexer, and it'll kind of break everything up and resuspend it. I can take a micropipette and pump it to mix it, right? So then I got my SDS buffers. I resuspended it, right here. Right, and I got my buffer in there, and then I'll I'll neutralize the pH of the lysate, right? And when I do that, I'll allow my plasmids to come back together. All right, so then I'll take this solution here, right, and I'll bind DNA from supernatant to matrix. So in this column, I have my resin beads, and that's my matrix. So I'll take this, and I'll put it into my column, right? And when I put it into my column, things that are not negative in charge will run straight through the column, right? And they'll end up in this supernatant. So now the supernatant's on the bottom, right? And now my DNA is caught in my beads. So my DNA here is trapped, right? I can use a wash buffer. So my wash buffer will remove things that are loosely bound to my gel. And you see where it says spin here? I forgot to mention this, where it says spin here. So this whole thing, my microcentrifuge with my microfilter will fill into a centrifuge. And it's spun to kind of pull things through, right? So I'll put my wash buffer in and I'll spin it. I'll put another wash buffer in and I'll spin it. And at this point, I should have pure DNA bound to that column, right? All the impurities should be gone, right? And then I'll eluent, right? So the eluent, I'll elute here. The eluent here is my final fluid that has my plasmids in it, right? So I'll use a wash buffer. So a wash buffer here usually has some salt. My elution buffer has a different type of salt, and it will pull all the rest. So once I get my wash buffer through, right, then I have purified plasmids. So here I have purified plasmids, right, and I'm ready to test them, right? So the above example wasn't as good as this because now I have just pure plasmids at the end, right? So DNA quantification, I can do it by gel, right? So if I know the size of my plasmid I use and I know the size of the DNA I put in there, I can run them through a gel and compare them to a molecular ladder, right? And I should have, in all my samples, I should be pretty consistent in my sizes. If I'm consistent in my sizes, then I know my mini prep and my quantification is pretty good. If I end up with different sizes, right, it could be two different things. Right, I'll use a molecular mass ruler. So the difference in plasmids could be because I may have plasmids that didn't come back together right. The distance traveled could be due to nicked plasmids, open plasmids. So remember, when I have a gel, and I have my well, and I have my DNA in my gel, in my well, right? Inside my gel is a matrix, right? So this is acting as a sieve, a filter. If I have a nick gel, right, which is single-stranded, that thing will just weave its way through, right? Or if I have a circular plasmid, it doesn't travel as far. Right? If I have a supercoiled plasmid, it's just tightly wound, it travels even less. So I can get different results based on whether I have twisted plasmid, mixed plasmid, open plasmids. So I can get different results. But if you look at the brighter bands on the gel, talk about greater concentration, right? They have to do with greater concentration. Right? And the last thing I can do is I can do a photospectrometry or spectrophotometry, right? And I could use a spectrophotometer to look for DNA, right? So I look for the higher the spike, the more concentrated it is. DNA absorbs at 260 nanometers of UV range, right? So they'll use a conformation, they'll use a protein that kind of shines at both sides and make sure that my spectrophotometer is working well. But this should be able to prove that I have DNA, right? I shouldn't have gDNA because that would have been a bigger band, but I can confirm it with spectrophotometry as well. All right, so pretty quick today. You have tests coming up. Make sure you review all your stuff, and I'll talk to you soon.